seminar, webinar. Last one of the year. We'll have our AGM in a couple of weeks, which we'll email everyone about very soon. But just to start off, I'd like to acknowledge the traditional owners and their custodianship of the lands on which we meet today, the many different lands on which we're meeting. On behalf of the ABSS organising committee, I pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. UQ, from where I'm based, has always been a space for teaching, learning, research and collaboration tens of thousands of years before it was established as a university campus, and this continues today. So thank you everyone for joining us for the talks by our winners, our award winners. Congratulations everybody. The committee that had to choose the winners had an incredibly difficult time because we had a lot of really excellent submissions, which made our lives difficult, but was also wonderful to see so many really outstanding contributions to the brain stimulation literature from Australasian students, ECRs and EMCRs. So that was incredibly heartening to see. So I'll hand over to Robin, who can introduce the first of our winners. We'll have the PhD winners up first of all. So over to you, Robin.
Great, thanks, Hannah. So first up, I'm delighted to introduce Jessie, who is currently in her third year of her PhD in the National Institute of Education, Nanyang Technological Institute in Singapore. Her background is in neuropsychology and cognitive neuroscience. Her research interest is to use different neuroscience modalities to look at the relationship between brain and behavior. And in today's talk, she will be sharing her recent publication on the effects of TDCS parameters on motor learning behavior.
Thank you, Robin. Let me just share my screen. Oops. Can you all see it? I'll just prep the laser pointer as well. All right, thanks, Robin, for the introduction again. And thank you, ABSS, for this award. So good afternoon, everyone. I'm Jessie, a third-year PhD candidate at the Nanyang Technological University, Singapore. I'm here today to talk about a recent publication by myself and our research team entitled A Meta-Analytical Review of Transcranial Direct Current Stimulation Parameters on Upper Limb Motor Learning in Healthy Older Adults and People with Parkinson's Disease. Oops. Okay, so I'm sure most of you here is well versed in this method called TDCS. So I will just briefly walk you through the slide. So TDCS is a wearable brain stimulator that applies current to two electrode patches known as the anode and cathode that's connected to the stimulator. So it is non-invasive, it's painless and lightweight. So the anode carries the positive charge to boost brain activity, while the cathode carries the negative charge to decrease brain activity. So in terms of learning, which is my area of interest, so a very simple way to explain this is if a brain area is injured or if it's impaired, and we want to boost that brain activity of that area, we will place the anodal electrode onto the scalp of that area that is impaired, to boost its function and to promote learning. So in our meta-analysis paper, we specifically looked at upper limb motor learning. So generally, motor learning here is defined as the process of acquiring new motor skills, or for older adults or patients like people with PD, it's relearning of these skills through repeated practice. So in research, we know from previous research that age-related and disease-related changes contribute to increased energy costs and reduce motor automaticity in these like older adults and people with PD. Hence, learning and performance deteriorates. So therefore, these population groups have to relearn these skills through repeated practice. So in research, upper limb motor learning performance is usually assessed via different tasks, such as learning to make like a series of keyboard button presses via what we call the serial reaction time task. There's also like the isometric grip force task, the pegboard task, writing tasks, or motor adaptation tasks. So the outcome measures are typically like the learning rate, the reaction time, and accuracy of this. So the aim of this paper is to synthesize available methodologies and results to guide future research on the usage of TDCS on upper limb motor learning in both these populations, healthy older adults and people with PD. The rationale for this is, up to date, many studies have been published on the positive effects of TDCS to improve motor learning performance. However, none have consolidated evidence on the effects of TDCS parameters. So what I mean by TDCS parameters are something like current intensity. So we do not know how strong the current should be to boost learning performance. We do not know the montage selection, like where should we place the anodal and cathodal electrode? Is it at the prefrontal cortex? Is it the motor cortex, etc.? We do not know the stimulation duration or the number of sessions. And it is not really known like what size of electrode should be used. So the size of the electrode actually affects the density of the TDCS. So our recent paper here has consolidated evidence on the effects of TDCS parameters. So we did a database search for studies that published from January 2000 to March this year. So all publications should be in English. We've searched through 3 databases, PubMed, Web of Science, and ProQuest. So we have 2 inclusion criteria over here. All studies should be either healthy older adults, age 60 and above, and people with PD. We need studies that have a cognitive task with upper limb motor behavioral learning as their outcome measures. So for our exclusion criteria, we do not want any studies that has other non-invasive brain stimulation other than TDCS. We do not want younger population less than 60 years old, and we do not want like pathological subject group other than PD. We also excluded studies that have more than one non-invasive brain stimulation technique to augment motor learning. So there are some studies that have included TMS first, then TDCS, so we exclude those studies. We also do not want studies that have other tasks involved other than upper limb. So there are quite a lot of tasks in Parkinson's disease that include like walking, gait, etc. that we exclude them. And last but not least, we exclude those studies that do not have a control group. So those studies that do not have a sham protocol included, we exclude those studies. So this is a Prisma flow diagram. We have finally included 32 studies. So we have also included a Pedro scale to assess the quality of each paper. So out of a full score of 11, a score of 9 to 10 is considered as excellent quality, while a score of 6 to 8 is regarded as good quality. So among our included studies, 18 were rated as excellent, while 14 were in the good classification. So the mean score, the total mean score for these 32 studies is 8.81, which indicates that good quality in the methods of the included studies. All right, so in these 32 studies, in total, we have 526 healthy older adults and 216 people with PD. We have extracted these TDCS parameters from all of these studies. We have also extracted like what upper limb motor learning tasks they have used. We have also then extracted the outcome measures, the behavioral outcome measures before and after TDCS intervention. And all of this are computed for meta-analysis. So this figure over here shows the overall effects of TDCS on upper limb motor learning. So we have grouped the studies into 2 populations, so the healthy older adults and the people with PD. So the horizontal lines here represent standardized mean differences of the intervention effects. So take a look at this line over here. So it's 0. So it means 0 standardized mean differences. So towards the right, it favors intervention, favors the TDCS intervention, and towards left favors control. So our meta-analysis here showed that there is significant positive effect for healthy older adults and people with PD, indicating that upper limb motor learning tasks did improve after active TDCS compared to sham stimulation in both of these populations. Okay, next, we have also conducted separate forest plots for each parameter that I've mentioned previously. So here I've summarized our forest plots in terms of intensity, montage, stimulation site, protocol, and duration. So let's take a look at intensity first. So in terms of intensity, out of 32 studies, 3 different intensities chosen. So 1 milliamp, 1.5 milliamp, and 2 milliamp. And in the brackets as well, I've indicated like how many number of studies out of the 32 have chosen 1 milliamp. So we can see that the majority here actually chooses 1 milliamp. Okay, so the ones highlighted in red are highly significant. So meaning that TDCS does improve learning in these parameter settings, the ones in red. So here we can actually see that most chose 1 milliamp, and indeed there is significant changes, so significant TDC effects. However, like 1.5 milliamp, 13%, so it's like only 3 studies out of 32 studies, and we can't really find any significant difference, most probably because very few studies chose like 1.5 milliamps. So similarly to like montage, most of them chose like the anodal stimulation. So the site is like targeting the motor cortex instead of other different areas. So for protocol as well, most studies chose online TDCS. So online TDCS here means that TDCS is applied and while participants are doing the task. So offline is TDCS is applied to them while they're at rest. So most studies chose the online protocol. Last but not least, duration as well. Most studies chose single session TDCS. All right, so in the last few slides over here, I would like to highlight some limitations and gaps in the current studies. So hopefully future research will be more cautious during planning of TDCS studies. So overall, there are 17 studies with older adults and 8 studies with PD patients reported significant positive or negative effects on motor performance after active TDCS compared to sham stimulation. So out of the studies, 2 reported negative TDCS effects with their chosen parameter, while all the others showed positive effects. However, one main limitation here is take a look at this funnel plot over here. So the distribution of the included studies was asymmetrical, so indicating that there is quite a significant publication bias. So the white dots over here are our included studies, while the black dots over here represents the imputed missing studies. So our analysis here showed that the adjusted effect size was actually lower compared to the observed effect size. So this means that there is greater likelihood that the studies in current literature, the TDCS studies in motor learning, in the current literature, reported positive results. So they're more likely to publish positive results instead of like the negative results, which is a very major limitation over here. So indeed, while I'm doing data extraction, I found that there is lack of published studies that report null or non-significant findings. So even if they did, it is just reported as like null effects with just the p-value stated. So there isn't any graphs or any further data which can be extracted for meta-analysis. So fair enough, yeah, it's biasing the positive results. Next, we were unable to draw definite conclusions as well as to whether specific stimulation parameter was beneficial for that motor learning task due to very small sample size, as I've mentioned previously. So when studies were categorized according to like protocols or their parameters, some categories ended up with very small sample sizes. So for example, when we categorize by stimulation site, so only 1 study applied TDCS over the premotor cortex, 2 studies applied it over the prefrontal cortex, and like up to 25 studies applied it over to the motor cortex. So due to this very small sample size, we cannot really draw definite conclusions whether which stimulation site is better than one another. We compare it. And under such circumstances, categories with small number of studies are likely to observe biased estimate of effect size. So future research may want to take this into consideration. So current TDCS research on motor learning lacks studies with large sample size, which could be found in other TDCS research on executive functioning, for example, like risk-taking. They have up to 170 participants, but for motor learning, it's only within 10 to 30 participants. So that's the difference in number of studies. Next is about the standardizing protocol or terms. So let's take a look. So for example, anodal stimulation. So studies have reported like the anode electrode is placed on the right M1, cathode is placed on the left orbital frontal cortex. So studies who use cathodal stimulation is just like swapping them. So cathode at the right M1, while the anode at the left orbital frontal cortex. But there are also studies that say we are using cathodal stimulation. However, this is their cathodal stimulation. So cathode is at the left M1, anode is the right orbital frontal cortex. So it is not clearly specified in these studies, like which cathodal stimulation is used. And it is not justified why is cathodal placed in either the ipsilateral or contralateral side of the brain. And because out of these 32 studies, only 5 cathodal stimulation and 3 studies used like the second type cathodal stimulation, 2 studies the third cathodal type of stimulation. So we cannot really draw like definite conclusions whether cathodal stimulation it's what's the effects of cathodal stimulation on motor learning behavior. All right, so this is my last slide. Overall, the take-home message here is that there is no optimal stimulation parameters were found, at least not in our specific choice of population. Different stimulation parameters in terms of intensity, montage, stimulation site, the protocol online or offline, and the duration could enhance upper limb motor learning in both older adults and PD. Future research on motor learning with larger sample size is warranted. Besides, future research should aim to elucidate the underpinning mechanisms of TDCS instead of just focusing on its efficacy and reproducibility. And this could be done by implementing like electric field simulation modeling and combining TDCS with neuroimaging techniques. So this is what actually my next research is. So we have combined like TDCS and FNIRS together to elucidate relationship between M1 and SMAE in motor learning with a simultaneous protocol. So if anyone's interested to discuss more about this multimodal protocol, we can do so in the Q&A or after this webinar. Yep, so thank you so much. That's it for my presentation.
Thanks so much, Jessie. Yeah, it was super interesting. I think we have a few minutes for questions, I think. Aaron, any thoughts?
Great presentation, Jessie. I might have missed this, but were they all just single session studies or did it include multiple sessions?
Yeah, we include single session and multiple sessions as well.
Okay.
So most studies did single sessions. Yeah.
Right. I'm just curious if you noted any difference between that, like if multiple sessions might have led to a stronger effect, or was that something that was hard to tell?
From here, you can see that both sessions are actually significantly different, meaning that both single session, multiple session, there is positive TDCS effects. However, we can also see that most studies use single session compared to multiple session. But there's one thing I would like to highlight here is single session, there's a duration between 10 minutes to 30 minutes. So we only see the significant effects in 10 minutes, but not the 30 minutes study. So meaning it's not that during that single session, the longer it is, the better it is. Yeah, it's not so. Yeah.
Great. Thank you.
Yep. Hannah, right.
Hey, Jessica, wonderful talk. Thank you.
Thank you.
Just wondering, I mean, I think it's super interesting, right? Because this is a bit out of my area, but I believe that a lot of recent work in healthy young adults is finding that there's inconsistent effects of TDCS, and there's actually a call to increase the intensity. And there was a recent preprint to show that actually increasing to up to 4 milliamps might get more robust effects in younger adults. And using, as you were suggesting, computational modeling to actually optimize electrode locations and things like that as well. So I was just wondering, given what you're finding in your meta-analysis and what the literature in young healthy adults is saying, what parameters you've chosen for your new study.
Okay, so because this one, as you can see, there's none using 4 milliamps, and it's because this is in older adults and PD patients. So fair enough, maybe 4 milliamps is slightly too strong for them.
Yeah.
So for my new studies, because we are actually wanting to compare between younger and older adults. So to standardize it, we actually chose the lower one. So we are not using the 4 milliamps.
Yeah, that makes a lot of sense. Yeah.
Yeah, just for safety issues, and we want to keep it consistent between the younger and older. So we just stick it. We are still deciding perhaps 1 milliamp or 2 milliamps, but in terms of what we have done here for our meta-analysis, it looks that 1 milliamp does give like quite a robust effect as well. So we will stick to maybe the lower intensity. And another reason that I would like to point here is because we are using the what's that called? The simultaneous TDCS apneas. So if you know that apneas, like we do have these optodes that are 3 centimeters apart. So the TDCS patch cannot be too big. So the size can only be very small, meaning that the density will be very high if you use 4 milliamps. So I think it's, we can't really use 4 milliamps for it.
Yeah,
so
that's great.
for safety issues, correct.
I can't wait to see what you find. It sounds very technically challenging and super interesting.
Yeah, thank you.
Did you see an interaction of like intensity by like anodal cathodal? So blood directional effects.
There is effects. I mean, anodal and cathodal. Is this what you mean?
Just whether I think there was some earlier work suggesting that if you increase the intensity too much for like the inhibitory form, you get like excitatory effects, more in terms of like motor plasticity, but yeah.
But similarly, increased intensity, then we have to also take a look at the density of it. So the electrode size must be slightly bigger. So it makes it impossible to do it with apneas to look at brain activity. So we have to like judge which is better.
Athena just had a question on the chat as well about if you looked at the effect of electrode size.
Yes, actually. So the electrode size is with the intensity. Okay, sorry that I did not present it here because we did not do the forest plot, but we did it in like a regression line. So I can just verbally tell you that. But the bigger the electrode size, it doesn't mean that it's better. Because those studies that use like bigger electrode size actually use like higher intensity, and those with like smaller electrode size use it with like lower intensity. So basically the density is about the same, so it doesn't really affect the intervention effect, if that makes sense. So for the 30, so let me just explain it one more time. So for the 32 studies, those with bigger electrode size actually chose like bigger intensity, and those with smaller electrode size use it small intensity. So basically the density between these two options are actually the same. So there isn't any significant intervention effect.
Cool. Thank you. I guess it might be time to move on to our next speaker.
Thank you.
So I'm excited to hear a talk by Lara, who will be talking about assessing cerebellar cortical connectivity using concurrent TMS and EEG. So this was a feasibility study. And just a little bit about Lara. So she recently completed her PhD in cognitive in the cognitive neuroscience unit of Deakin University. Is now working as a postdoctoral research fellow in the Department of Neuroscience at Monash University. Her PhD focused on non-invasive stimulation of the cerebellum and the cerebellum's role in neurodevelopmental disorders. She's currently using diffusion-weighted imaging look at free water in the brains of individuals with cerebellar ataxia. In the future, she would like to explore how we can better tailor TMS to the cerebellum and combine cerebellar TMS with neuroimaging measures. Over to you, Lara.
All right, thank you. I'll just share my screen. All right, can everyone see that okay?
Yep, that's perfect. Thank you.
Great. Okay, thank you very much. Firstly, ABSS for this fantastic opportunity and their generosity, and also to the other co-authors of this paper. Okay, so I'll just take you through a brief overview. So I'll just first talk about the cerebellum, what it is, and how it is connected with the cerebral cortex. I'll also then talk about just very generally cerebellar non-invasive stimulation, why we do it, and some of the caveats associated with it. And then I'll discuss the study, and then follow with a very brief discussion and conclusion. All right, it's being a bit slow. There we go. All right, so the cerebellum, what is this? So most of you would know that the cerebellum is a subcortical structure, but what is less widely known is that it has distinct motor and cognitive regions. And these regions are all connected to the cerebral cortex in various ways. And these cerebral connections are very important. Efferent connections with the cerebellum, connections that exit the cerebellum going to the cerebral cortex go through the thalamus, called the cerebello-thalamo-cortico tract, and afferent connections, so connections coming back into the cerebellum via the from the cerebral cortex, go through the pontine nucleus. And these connections are very key in the formation of forward models in our brain. So this assists us with motor planning, the production of smooth movements, adaptive learning, and also more recently, these connections have been shown to be very important with cognitive functioning. So for instance, sequence learning, working memory, executive control, attention, and even emotion regulation. So just very briefly, I'll just give you a brief history of cerebellar TMS studies. Historically, the most widely used cerebellar stimulation protocol tests a thing called cerebellar brain inhibition, or CBI. And here a single pulse is sent to the cerebellar cortex to activate cerebellar Purkinje cells. And this is typically done with a double cone coil, or DC coil, because this coil just has a lot greater depth range and has been shown by past studies to be more reliable than flat coils. So now, because the output signal of cerebellar Purkinje cells to cerebral sites are inhibitory, this results in potentials evoked by a second pulse to the M1, which many of you can be measured by a motor evoked potential taken from a peripheral muscle, is reduced, as we can see in this graph to the far right. But what if we want to test CBI on non-motor sites which have no obvious peripheral outputs? And this is where TMS EEG comes in, where we can use the EEG signal to assess cortical potentials at a variety of non-motor sites. And past cerebellar TMS EEG studies, however, have used very low stimulation intensities, which have often been thresholded on the cerebral cortex. And the cerebral cortex has a very different cellular structure to the cerebellum, so we don't actually know what these studies were measuring. They also didn't have very stringent artifact control methods, which is very important in TMS EEG studies, because there's just a humongous amount of artifact that is generated in the EEG trace following a single TMS pulse. So in response to these shortcomings, I teamed up with Nigel Rogash and Mana Bayabani, who are TMS EEG experts, and we decided to look at what is the feasibility of conducting valid studies using single pulse TMS to the cerebellum with concurrent EEG. So we wanted to know how noisy the EEG signal is when applying TMS at suprathreshold intensities. And also, can we use current post-processing methods to salvage any of the true signal? So our design was, we just started with 10 healthy adults, just a very small sample. We just wanted to test the feasibility of doing this. We applied 80 single pulses to the right cerebellar hemisphere at 60% MSO, which has been with a DC coil, it's been shown by a previous study of ours to at least, it's the lower bound for eliciting CBI. We also used 90% MSO with a flat figure-of-8 coil, which is essentially gives us very similar depth characteristics at that intensity to the DC coil. And we also applied concurrent EEG during these trials. We also had some site controls. We had 80 single pulses to an occipital parietal site using both coils, and we tested phosphenes in both cases for phosphenes.
So it looks like we've lost our speaker. Maybe we'll just give her a minute to reconnect. Alex, do you think maybe we should move on to the next talk, or? And then
yeah, we can maybe move on to say Ono for now, and then if we get connection back, she can just go after Ono, if that's okay with you, Ono, to go ahead.
Yeah, that sounds like a good plan.
Yeah, okay, no worries, thanks so much. So thank you, everybody. We will hopefully get the speaker back, but in the meantime, we'll move on to our next presentation, which is our ECR presentation from Dr. Ono van der Groen. Ono is actually a research fellow in Perth at Cowan University, and he uses non-invasive brain stimulation to enhance human performance. He did his PhD at ETH in Zurich, and now where he focused on transcranial random noise stimulation. So today, Ono is going to give us a talk about TRNS. So thank you, Ono. When you're ready, go for it.
Right, thank you. All right, great. Well, thanks for having me, and thank you for the award. First of all, so I can see my slides. So today I will talk to you about the effects of TRNS on brain and behavior. So this is from a paper, a narrative review that got published this year. This was actually one of the papers that I wrote during the COVID lockdown. There were the multiple COVID lockdowns we had here. So this paper is a narrative review which gives an overview of the effects of TRNS on human perception, cognition, and learning. It also explores the mechanisms that might underlie the effects of TRNS on these functions. So TRNS is a relatively new brain stimulation method. So the first publication with TRNS was in 2008. So there's not too many publications on this topic yet. So first of all, what is TRNS? So transcranial random noise stimulation is an alternating current stimulation with frequencies ranging from 0.1 to 640 Hertz. So in the literature, people usually talk about low frequency stimulation, so 0.1 to 100 Hertz, and high frequency stimulation, 100 to 640 Hertz. So this cutoff of 100 Hertz seems to be a little bit arbitrary. And the higher end of the frequency spectrum is 640 Hertz. And in our lab at the time, this also had a practical reason because of the limited sampling rate of our TCS device as well. So TRNS is zero mean Gaussian white noise. So zero mean is what you can see in the second figure here. So the TRNS intensity of 1.5 milliamp on average. We don't apply any current. The average would be 0 milliamp. And white noise means that there's a constant power across the different frequencies. So here you can see 100 to 640 Hertz with a constant power. So TRNS is non-directional, so there's absence of a DC field. However, some studies have added a DC field to TRNS to investigate the effects of this. And it's also polarity independent, so it's not anodal or not cathodal stimulation. So some benefits of TRNS that have been mentioned in the literature, it's that it can have larger behavioral after effects. It's less affected by cortical folding because there's no DC current, so this might make it easier or more easy to apply as well. And with TDCS, some currents cancel each other out. With TRNS, this does not happen because it depolarizes regardless of the current flow orientation. So these could all be benefits of TRNS compared to, for example, TDCS. So I will mainly talk about online and offline effects. So just for your understanding, so online is when we apply TRNS during a task. So we can either look at acute effects, so the acute effects on behavior, or also chronic effects, so where we apply TRNS during a learning task, and we look at the behavioral effects before or after. And then we also have offline effects, where we, for example, look at neurophysiological effects, apply TRNS during rest, so people just sit around to look at a fixation cross, and then we apply TMS again to look at neurophysiological changes. So this is, this must be the first paper applying TRNS. So they applied 10 minutes of TRNS targeting the primary motor cortex, and they could see an increase in cortical excitability measured with TMS. And this effect lasted for 60 minutes after stimulation. And after 90 minutes after stimulation, this effect was not there anymore. So this finding is pretty robust because it has been replicated a few times. So there's one study which also looked at, so there are a few factors that influence TRNS effects on excitability. So these are two studies that have been conducted. So one in 2012, where they showed that 1 milliamp can indeed enhance excitability, but 0.4 milliamp can decrease excitability. And this decrease in excitability only occurred 20 minutes after the stimulation was stopped. There's also an effect of frequency. So where there was no effect on cortical excitability, if you stimulate between 400 Hertz and 400 to 700 Hertz, but you do see effects if you use the whole frequency band. So what do we know actually about the neurophysiological effects of TRNS? So we know that it involves sodium channels. For example, sodium channel blockers reduce the TRNS-induced excitability changes that we've just seen before. And also adding noise to cells in vitro causes an acute repetitive opening of sodium channels as well. So this could be one of the mechanisms underlying the effects of TRNS. So where there's continuous opening could result in membrane depolarization and could induce LTP-like changes. So there's also another effect or other underlying working mechanism that has been proposed. which is called stochastic resonance. So with stochastic resonance, the idea is if you have a weak signal which does not cross a threshold in a nonlinear system, you add a little bit of noise. The input comes a little bit closer to your system threshold. When you apply an optimal level of noise, the input actually crosses the threshold and you'll be able to detect the signal. When you add too much noise, the signal gets buried in the noise. So here you can see the relationship between noise intensity and the signal to noise ratio. So where there's will be an optimal noise intensity which enhances the signal to noise ratio. So this is one of the other possible underlying effects of TRNS. So there's a publication in 2018 as well, which looked at the different mechanisms that could underlie acute effects of non-invasive brain stimulation, which are stochastic resonance, rhythm resonance, network entrainment, and even imposed brain patterns. Where you could see on this axis the field intensity needed to induce these kinds of effects. And stochastic resonance is one of the effects where you need the smallest amount of induced current in order to get this effect. So probably with TRNS, we might not be able to impose any patterns or do any network entrainment, also because the intensity that we apply is way too little for this. There's also an involvement of GABA. So TRNS-induced plasticity is GABA receptor sensitive. And also a recent study showed that these levels can be reduced after prolonged stimulation in juvenile mice. So prolonged stimulation in this case was 9 sessions of 20 minutes spaced over 5 weeks. So it wasn't even that long. So recently we were thinking we should have a look at acute effects of TRNS in humans as well. So this is a study that we completed in Zurich, where we applied combined TMS with TRNS. So we applied TRNS targeting the primary motor cortex. During TRNS application, we looked at cortical excitability changes. So this is the proposed model that we have, where there will be neurons that are non-responsive. There will be neurons that are responsive toward TMS stimulation. And there will also be neurons that are responsive during the TRNS. So which you can see here, which are the blue ones with the orange circles around it. So what we did find is that when we apply TRNS, the probability of eliciting an MEP is significantly increased. So this is the data of a typical subject. When we have no TRNS, the intensity of TMS, when we have no TRNS, the TMS intensity that we need to get a MEP with a probability of 0.5 is about 37.8% MSO, maximum stimulator output. Whereas when we apply TRNS, this goes down to 36.7 roughly in this case. So these are very small effects, but it was very, very robust across all the participants that were tested. So we also know that TRNS has acute effects on perception, which has been replicated a few times. So this is one of the studies that we run where we apply TRNS during a visual detection task. And you could see that the correct responses, so the performance in this task significantly increased when we applied TRNS. So this did not happen, for example, when we applied TRNS to a control condition. Acute effects of TRNS have also been observed so by others in the visual system and also in the auditory system as well. So this was just the studies before. They were just on perceptual decision making, so like a detection task. So we also know, which has been replicated a few times as well in the literature, that TRNS can affect perceptual decision making. So the usual task that we use is then motion coherence tasks, where people have to indicate if the global motion of the dots is going to the left or to the right. So the 100% coherence. So this one goes to the left. The 3% coherence, I'm not quite sure because I can't really see it at the moment. But what we find with TRNS is that the performance on this task also significantly increases when the task is very difficult. So this is what you can see here. So the performance, when we apply a small amount of noise, so 0.25 milliamps of TRNS, we could see an increase in performance in this task. When we did some modeling on this, we could see did a drift diffusion model, we could see that the drift rate significantly increased when we applied a little bit of TRNS, which suggests that the quality of the sensory information has improved by applying TRNS. So these effects on perceptual decision making on this task have been replicated by a few different groups as well. So this seems to be quite robust. So we also have TRNS effects on learning. So this was one of the first tasks that looked at this. So they had a perceptual learning task, which consists of 7 blocks. It's an orientation discrimination task, so people have to indicate if the first one or the second target is tilted more towards the left or to the right. So here you can see that over the blocks, people who received high frequency TRNS had a much better performance. So the slope of this graph, which is the learning rate, was higher after high frequency TRNS compared to low frequency TRNS, TDCS, and sham stimulation. These effects of TRNS on learning can also last for quite a long time. So this is a very nice study in 2019, which showed that 10 days of training on a global direction motion test for 20 minutes a day with TRNS could significantly improve the performance on this task. And the speed of this perception, so it could increase the performance on this task, and these effects were still present 6 months follow-up as well. So here's a measure of amount of learning. So 6 months following this task. So participants came back to the lab and they still could see an improvement on this task compared to people who didn't get TRNS or who received TDCS. And the nice thing of the study is, as well, is that it has shows effect in healthy controls and also patients with chronic cortical blindness as well. So we have all these effects on behavior. So what do we know about some neural correlates? So a few studies have looked at this. So one study in 2017 showed that when there were near threshold auditory stimuli, there was a reduction in the peak latency of auditory ERPs. And this suggests that TRNS might facilitate the firing of neurons involved in this task, which then resulted in this reduction in peak latency of the ERPs. Another study looked at the effects of TRNS on sustained attention, and before and after this task, they looked at theta-beta ratios, which are thought to reflect top-down attention. So they could see that that there was a reduction in this ratio, which coincided with behavioral performance. Then 5 days of training on a complex task coupled with TRNS, targeting the dorsolateral prefrontal cortex, led to more efficient neurovascular coupling. And a recent study, which was quite nice, where they applied TRNS over the parietal cortex with training on attention tasks, showed an increased connectivity in dorsal and ventral attention networks. So they could show for the first time that TRNS effects can have network-wide effects, and which correlate with the performance improvements of this specific task. So therefore, there are a few studies which suggest that TRNS can lead to more efficient neural processing. So there are a range of different factors that can influence TRNS effects, which might not be specific to TRNS, but might also apply to brain stimulation in general. For example, people who already performed very good at baseline might not benefit as much from TRNS. And another question might then be, they might not benefit as much in their performance, but they might be able to sustain their performance for longer. But as far as I know, nobody looked at this with TRNS. There's also an age effect. So some studies found that TRNS or face memory can be beneficial in young adults, but not in elderly people. So it might also be that high performers also, they show to recruit different brain regions as well. So if you have a TRNS experiment and you just apply the TRNS to brain regions that you think are on average involved in this task, then you might not see effects anyway. So therefore, probably moving to a more individualized approach of TRNS might show larger effects. Okay, so what we did find was that the TRNS can have direct online effects on perception, and these effects are quite robust. So there are different studies who have shown these effects. It can also improve learning. The exact working mechanism is unclear, but there's a role of sodium and a role of GABA as well. And one of the underlying mechanisms could be stochastic resonance. So then the last question is then, well, is TRNS really that special? So that's so there was a new PhD student who started in the lab in Zurich after I left. So we looked at a study where we used this visual contrast detection task, and we applied different types of TCS. So this study started because I was contacted by a mathematician who said, well, you applied random noise, but on a mathematical basis, the effect should be larger if you apply triangular waveforms instead of random noise. So then we thought, okay, let's try this. Let's see what's happening. So we applied TACS triangular waveforms, TACS sine waveforms, and we compared it to the effects and contrast detection of high frequency TRNS. And we could see that all of these brain stimulation methods improved visual contrast modulation, and TRNS was not superior compared to high frequency TACS or high frequency triangular waveforms. So TRNS, can be beneficial, but other waveforms, which include high frequency waveforms as well, might show similar effects. So then finally, my acknowledgements. So this paper I've written together with Veronica Nicol from the ETH in Zurich, Jason Mattingly from the Queensland Brain Institute, Grace Edwards, who was at Harvard, the School of Psychology in Harvard, and Dylan Edwards, who is a professor at ECU and a director of the Moss Rehabilitation Research Institute in Philadelphia. So that was my talk. Thank you all.
Great, thanks, Arno. That was really great. I think we have probably maybe time. Do we have time for some quick questions, Robin? Do you think?
I think so. Yeah.
Does anyone have any questions? You can put it in the chat or raise your hand. In the meantime, I might ask you a quick question, Arno, because I'm obviously very interested in the mechanisms, and you showed some interesting things on the voltage-gated sodium channels. So I was just curious on how they think what is TRNS actually doing to the voltage-gated sodium channels to have that kind of effect on excitability?
So when you apply TRNS, you get repetitive opening of the voltage-gated sodium channels. So there will be some sodium influx. So there will be a bit of depolarization. So that's what they found in the studies.
I was curious, like why that might be different, between TACS and TRNS, because presumably both are repetitively opening or depolarizing the voltage-gated sodium channel. So, but why TRNS seems to do that more so, or the evidence seems to be more so for TRNS than for, say, TACS?
Well, I used to be a very, very strong believer of TRNS because I did my PhD on this. But then after the last study that we run, where we had high frequency TACS, which was 80 Hertz, we could also show similar behavioral effects. So I don't know if anybody ever looked at the effect of 80 Hertz high frequency TACS on voltage-gated sodium channels. So it could be actually that it doesn't matter what you use, as long as it's a high frequency signal. So 80 Hertz maybe for TACS, or also the triangular waveforms that we used as well. At the behavioral level, they have the same effect, but I don't know if they have it in a physiological level, if they have the same effect as well.
Cool. Any other questions? No. Okay. So in the interest of time, we might just move on quickly. Thanks so much, Arno. That was really interesting. And thanks for moving your talk up a bit earlier. So we will go back to Lara, who had a power cut in the middle of her talk, but she's back now. So Lara, if you can share your slides again.
Yeah, sorry everyone. Yeah, it was so windy here that the power went out. So I think, right. I think I was here on the design. Okay, so I'll move straight into this. So yeah, we had 80, I think I went through, we had single pulses to the right cerebellar hemisphere with both coils. We also had a site control over the occipital parietal regions, again, using both coils. And importantly, we also included a multi-sensory control, which was an electrical stimulus used to mimic the twitch sensation of the TMS pulse, because TMS over the cerebellum is very uncomfortable. It does elicit a lot of twitch because it really triggers muscles around the base of the neck and also the face. And so we needed to sort of mimic as a control measure, those sensory outputs coming from the TMS. We also used a concurrent TMS coil discharge with the coil angled away from the back of the head. So it didn't actually activate the brain tissue, but we could still get that auditory output from the TMS coil. And we also pressed the coil against like a plastic spacer held against the back of the head to get some vibration effects of the coil too. And in all conditions, we had auditory masking. And we also gave the participants a visual analog scale to measure their level of discomfort, pain, twitch response, and the amount of masking in each condition. Okay, so for data processing, we used the Tessa MATLAB toolbox, which is a dedicated TMS EEG data processing package developed by Nigel. And initially we used a cleaning pipeline tailored for reducing artifact from single pulse TMS applied for the M1 using a figure of 8 coil. But due to the large artifact that we were seeing from the cerebellar stimulus, we introduced the sound filtering algorithm prior to the ICA step to see if this would improve the final signal. And then to see if we could reduce the influence of sensory potentials even further, we used an approach taken by Mana in an earlier study, which applied SSP-SIR to our TMS outputs. And SSP-SIR takes a known artifactal signal, in our case, the trace from the multisensory control, and removes it from the signal of interest to leave just those non-sensory potentials evoked by the TMS. So just now looking at some outcomes, for the VAS scales, we didn't find any difference in pain and masking scales across the conditions, which is really good. For discomfort, we did see that the double cone coil was more uncomfortable than the figure of 8 coil, which most people would say, yes, it is a lot more uncomfortable. As for the twitch response, the double cone coil did elicit a higher twitch response than the figure of 8 across both sites. And that's because the double cone coil is just larger than the figure of 8 coil, and it does tend to wrap around to the face, the jaw muscles a lot more. And generally the double cone coil was more uncomfortable than the figure of 8 coil, particularly at the cerebellar sites, most likely due to that large twitch response. Okay, so let's look at some of the actual EEG outputs. So we can see in the first raw image that the amplitude of the raw signals were very large in all conditions, particularly when the double cone coil was applied over the cerebellar site. So that's the first graph in each A, B, and C. Channels also had very long recovery times. We did notice that the amplitudes of the electrical control were significantly less than for the CBDC condition, which may have indicated that the full extent of the sensory potentials were not fully accounted for in our control method. We go onto the image on the right. We can also see that processing the signal using Tessa and sound filtering substantially improve the signal by reducing noise in the conditions at latencies prior to around 60 milliseconds, which is that's the time range that we're interested in, the time range that's most likely corresponding to the brain activity of interest. Then the signals became more similar and centralized after that, consistent with the N100 which is a potential typically associated with sensory activity. So to explore the peaks at these early latencies further, we used the Spearman's rank correlation of the grand average signals, and we took spatial correlations, which are the correlations across electrodes for each time point, and the temporal correlations, which are correlations across time points for each electrode, of each of the TMS conditions with the multisensory control, just to see how similar the signal profiles would be to the multisensory control. And we found strong spatial correlations as early as 48 milliseconds post-pulse in all conditions, but the temporal correlations tended to be highest at mid to late latencies, which again were consistent with sensory artifacts. And interesting, the lowest correlation between the cerebellar DC condition and multisensory control conditions was at a contralateral posterior parietal site, which is this kind of bluish region here, which has known connectivity with the cerebellum. So then to see if we had any effects of coil type and stimulation site, we compared conditions using cluster-based permutations with paired sample t-tests over the first 60 milliseconds. So firstly, when we compared the cerebellar DC condition against the multisensory control, we saw a significant difference in a contralateral posterior parietal site at around 40 milliseconds. DC condition, so that's across sites for the DC coil. We also saw a difference over parietal electrodes Sorry, did that cut out again?
Sorry, Lara. Yeah, we lost you.
Oh, sorry. So we then compared the double cone.
Might just want to screen share just the connection.
Oh.
Lara, did you want to try stopping your video as well? It might just help with the speed a little bit.
That's a good idea.
Yeah. Okay. All right. Okay. So we then we looked at the outcomes of the figure of 8 coil, and the cerebellar figure of 8 coil condition was quite similar to the multisensory control. And we didn't see much difference between the CBF8 and OPF8 conditions, which suggested that much of the response from the F8 coil was dominated by the sensory components. And finally, when we compared the double cone to the F8 coil at the cerebellar site, we also saw a larger peak in the CBDC condition between 41 to 60 milliseconds that approached significance. So that was, I think, about P equals 0.03. Which further supported the possibility that the CBDC condition evoked a component over posterior parietal sites through cerebellar thalamocortical pathways. So now just focusing on the cerebellar DC condition with SSP-SIR. So following the SSP-SIR filtering, we noticed that activity over occipital parietal sites was heavily suppressed, but the most prominent peaks were still present, as we can see in the bottom image in figure A. That's being zoomed in to show those early peaks. The voltages were very much reduced though, And notably from the corresponding source estimates, the contralateral posterior parietal component that we saw in the cluster-based permutations was preserved. Where this is promising, but unfortunately, because of the large amount of artifacts that we saw in the raw trace and the amount of post-processing we had to do to get to this point, this evidence is still somewhat inconclusive. So we don't know if the EEG is actually picking up the true signal of cerebellar connectivity with the posterior parietal cortex. So what are the implications of this study? Well, there's obviously a large amount of artifact generated in the EEG trace when stimulating the cerebellum at suprathreshold intensities, and these can come from a range of sources. And this means that future studies have to be very careful about the artifact control methods used within sessions, and also be mindful that a high degree of post-processing will be required. We do think that the double cone coil overall is preferable to the F8 coil, despite the discomfort differences to deliver the cerebellar stimulus, because historically it's more reliable, and due to the trace from the figure of 8 coil being quite similar to the multisensory control, even at early latencies, I think it's just going to be less reliable. However, we can't be sure. I'll just reiterate that the parietal component we observed from the CBDC condition was a true component due to the large amount of noise present in the original trace. So ultimately, for cerebellar TMS EEG to move forward with any confidence, I think studies have to use, say, quiet coils to reduce the noise output. And also, we need to develop cerebellar-specific stimulation methods, including coils that can better target cerebellar sites at lower stimulation intensities. And this is definitely something to think about for the future development of TMS equipment more generally, but also how we design and implement cerebellar TMS studies in the future. Right, well, thank you very much for listening, and apologies for the electrical disruptions. And thank you very much to ABSS for this opportunity, and all the co-authors. I just have a special mention to my supervisor, Pete Endicott, and also Dr. Michael Doe, who helped me a lot with the equipment setup, and of course, Dr. Nigel Rogash and Dr. Mana Baibani.
Thanks so much, Lara, for persevering. That was really interesting. Do we have any questions from the audience? No.
Not so much a question, but I just wanted to say thank you so much for that talk, Lara. And I mean, this seems like really important work to be doing in the space of combined stimulation and EEG. So, you know, really great contribution. It's awesome.
Thank you, everyone.
Alex, did you want to take over?
Yeah, sure. If there are no other questions, you don't have anything else, Robin, then we will move on to our final speaker, who is Robin himself, which is why I am chairing the second half. Robin is a research fellow at Monash, and his research looks at trying to optimize brain stimulation for the use of psychiatric disorders. So, Robin, I'm sure you take it away when you're ready.
Okay, yeah, just to clarify, I'm at the University of Melbourne, but I was at Monash. Can you guys see the slides all right?
No, I can't see anything.
No, it's not sharing at the moment.
Oh, of course. Okay. Thanks for that. All right, so I'm just going to present a brief overview of my research on targeting neural circuit dysfunction using brain stimulation. And this approach aims to optimize TMS therapeutic outcomes by aligning brain stimulation targets with disorder-specific brain network architecture. So I'll mainly be talking about this paper where we develop methodology to enable personalized circuit-based targeting. And I'll also describe work where we retrospectively validated the importance of this approach in a clinical cohort, and then move on to introduce some of our recent research applications. So the most common therapeutic application of TMS is for treatment-resistant depression, where it is typically targeted to the dorsolateral prefrontal cortex. And over the course of several weeks of daily treatment, some people respond very well, but many do not. And overall, the response rate is around 35 to 50%. So this is not bad, considering this is a treatment-resistant population, but nonetheless leaves a lot of room for improvement. And one basis for this variation may be clinical or demographic factors, although these have shown somewhat weak and inconsistent effects across studies. Another potential factor is suboptimal spatial targeting. However, over the past 2 decades, the optimal TMS target site has remained elusive. A range of targeting methodologies have been explored, and you can see here that these have targeted quite different subregions of the dorsolateral prefrontal cortex. And without going into detail, recent evidence indicates that connectivity-based targeting strategies, and in particular, targeting strategies that are informed by an individual's brain connectivity, show the most robust relation to TMS therapeutic outcomes. So which connections should we be targeting? Several studies have shown that TMS treatment outcome relates closely to functional brain connectivity between the DLPFC stimulation site and a deeper brain region known as the subgenual cingulate cortex. This provides an obvious opportunity to improve treatment outcomes by prospectively selecting the DLPFC stimulation target based on optimal connectivity with the SGC. The easiest approach would be to identify a single one-size-fits-all DLPFC target site that should work for all individuals, defined by a group average map of subgenual cingulate cortex functional connectivity across the DLPFC. It is worth adding that the initial reason for focusing on the SGC is that this region was consistently implicated in antidepressant response across a range of different therapies. And on this basis, the SGC was also selected as the target region for deep brain stimulation. Potential disadvantage of a one-size-fits-all targeting approach is that there is substantial inter-individual variation in brain network architecture, particularly within the prefrontal cortex, as highlighted here in hot colors, yellow and orange, corresponding to increased evolutionary development within this brain region. These areas can vary to such an extent that individuals can be reliably identified from a large sample based on their functional connectivity fingerprint. And critically, a few studies have delivered TMS to a singular one-size-fits-all group target using neuronavigation, but haven't shown any compelling improvement in treatment outcomes relative to conventional targeting. So together, these findings provide a strong rationale for personalization of the DLPFC target site and defining this site according to connectivity with the subgenual cingulate cortex. But in order to translate this approach clinically, we first need to know whether precise personalized targeting is even feasible. If personalized targeting based on brain connectivity is feasible, then it should be possible to reliably identify personalized targets that remain near identical in position across 2 different brain scans. However, when we started this work, a paper had just been published using conventional methodology, indicating that targets differed by an average of 3.5 to 4 centimeters between 2 separate scans for a given individual, even when using the best quality brain scans from the Human Connectome project. In other words, personalized targets were not reproducible and personalization did not appear to be feasible. So without going into detail, we developed computational personalization methodology, and this ultimately enabled us to precisely and reliably pinpoint optimal personalized connectivity-based stimulation targets. We verified in 1,000 individuals from the Human Connectome project that the sites we identified varied by only 2 millimeters or a single brain voxel between scans repeated the next day or even 1 year apart. Importantly, these personalized targets were not only robust, but also differed substantially between individuals, demonstrating that our approach preserved inter-individual differences. Interestingly, we also had familial data from the Human Connectome project and found that targets were more similar between monozygotic twins compared to dizygotic twins, non-twin siblings, and unrelated individuals, suggesting that these targets are stable and under genetic control. So with this approach, we see an increase in the reproducibility of personalized coordinates, individual-specific rather than generic coordinates, and a striking increase in the spatial correlation of the SGC functional connectivity map across the DLPFC region when compared across two separate scans. So for a 15-minute scan, conventional methodology gives us a spatial correlation of 0.2, whereas with our approach, this increases to 0.9. We then tested whether personalized targeting is clinically meaningful in a sample of 26 individuals who had previously undergone a brain scan and who received TMS for depression, and for whom the stimulation target and treatment outcome was known. We retrospectively computed the optimal personalized stimulation target from each individual's brain scan. We anticipated that individuals whose actual stimulation target was by chance closer to their optimal stimulation target would have a better treatment outcome. And here you can see that a scatter of optimal personalized stimulation target sites across this cohort. And here you can see that our data confirm this hypothesis. So this figure shows that individuals who serendipitously received TMS closer to the optimal stimulation site consistently had better treatment outcomes. Moreover, these findings have since been independently replicated across two separate cohorts. In our paper, we also showed that there was no relation to TMS clinical outcome when we instead computed proximity to any of the one-size-fits-all group targets that I showed you earlier. In parallel, other groups have been working on this, and there is now preliminary prospective evidence emerging that connectivity-based personalized targeting may enhance clinical outcomes across invasive and non-invasive brain stimulation modalities. Across 6 separate but small studies, including an RCT just published in the American Journal of Psychiatry, the average response rate is 87%, and the average remission rate is around 65%. This is vastly higher than with conventional targeting, where the average response rate is 46% and the remission rate is 31%. These findings seem very promising and clearly warrant larger clinical trials. The importance of brain connectivity in mediating clinical outcomes is also becoming increasingly evident with another form of brain stimulation, known as deep brain stimulation, where preliminary evidence from Helen Mayberg's group indicates a response rate of 81% with personalized connectivity-based targeting, compared to 41% for more conventional anatomical-based targeting. So since publication, there's been uptake of our methodology at various sites worldwide, and we've developed a toolbox to facilitate clinical translation. Our approach has been TGA approved, and our collaborators launched one of the first clinics in the world to offer personalized TMS with promising results to date. We're also undertaking a pilot trial in adolescent depression at origin. So the next key question is whether the benefits of personalization based on functional connectivity extend to other brain regions and TMS applications. For instance, previous work has shown that multi-day TMS to the parietal cortex can enhance associative memory performance, the parietal cortex being a key component of the cortical hippocampal memory network. As you might have noticed before, the parietal cortex is another brain region that has high inter-individual variation in brain connectivity. So without going into detail, we retrospectively analyzed an existing cohort where MRIs were acquired and this multi-day TMS protocol was delivered. We knew the actual stimulation coordinates and applied our personalization methodology to compute individual-specific optimized target coordinates. And these data showed once more that closer proximity to these optimal personalized targets was associated with better TMS-evoked improvements memory performance, indicating that the benefits of personalized targeting generalize beyond just the prefrontal cortex. In this slide, I wanted to just quickly make the point that TMS tends to impact and modulate distributed brain networks rather than a single brain region, such as the local stimulation target site. So here, following stimulation of the cerebellum, you can see changes in brain connectivity occurring with real but not sham TMS across different regions in this cognitive network, but with no change at the control site. Other research has indicated that the most therapeutically effective TMS and DBS targets, as well as lesions eliciting symptoms, are functionally connected to the same disorder-specific brain circuit. And last year, our collaborators demonstrated this to be the case in depression, delineating what they have termed to be a convergent brain circuit for depression. This becomes relevant, as I'll show you on this slide. So a recent meta-analysis demonstrated that across the past 20 years of task-based imaging research into, across the past 20 years of task-based imaging research into emotional and cognitive dysfunction in depression, comprising almost 100 neuroimaging studies, there was no convergence in terms of the anatomical loci that demonstrated aberrant activity during these tasks. This finding raised alarm bells in the field around the reproducibility of research findings. The lack of convergence might be partially attributed to the incentive for novel task designs across different studies, small sample sizes, and other factors. In my latest research, I considered whether these regions might be linked or functionally related from a connectomics point of view. So I'll skip the details of the methods and null conditions, but in short, these divergent coordinates actually localize to statistically highly robust distributed brain networks. And importantly, these networks recapitulate clinically meaningful and independently derived models of depression circuitry. Moreover, when I use these networks as a template to define personalized targets in the same way I described earlier, TMS therapeutic outcome was dependent on how effectively this emotional dysfunction circuit was targeted. So in sum, these findings provide highly novel and independent evidence for a convergent brain circuit in depression, and it appears that more effective personalized targeting of this convergent circuit leads to amelioration of depression symptoms. One other thing, so in our group, one of the students in our group has been working on defining a neural circuit target for obsessive compulsive disorder and determining brain stimulation target sites that might be most clinically effective. And lastly, I just wanted to thank Andrew Selesky and all those who have collaborated and shared data with us.
Great, thanks, Robin. Do we have any questions for Robin?
I have one, Robin. Wonderful talk, thank you. Just wondering, what do you think is the next step in this space? Have you considered other individual difference factors that you could also include maybe to try and increase efficiency or effectivity of this treatment? So something like baseline neurochemicals or other baseline measures in participants?
Yeah, great question. I think from our point of view, I guess we want to start by prospectively validating these findings. So everything so far is retrospective. So really we would like to start with depression and just prospectively validate that this does actually improve treatment outcomes before then kind of moving on potentially to other disorders where we can apply the exact same framework. Yeah, I guess I would probably, in terms of neurochemicals and that kind of thing, I might leave that to other people who have more expertise in that area. But yeah. I think, yeah, it's kind of, I guess, in a sense, kind of baby steps, but it's kind of, yeah, validation, prospective validation at this point would be the main priority.
Yep, not baby steps at all. Sounds really important.
Yeah.
Robin, we have a question in the chat from Athena. So she says, just wondering if the OptiStim toolbox is something that is publicly available, and if so, where it can be found.
Yeah, that's a good question. So it's not yet available publicly. We actually wanted to validate that toolbox in a clinical trial that we're trying to get funding for. Yeah, but I mean, if you want, Athena, yeah, get in touch and we can discuss sharing it. So it makes the whole thing a lot more user friendly. So people don't even need to really understand code. It's just kind of you input the MRI image, or you point the toolbox to the MRI image. It uploads it, preprocesses everything, figures out the optimal coordinate, and basically that's the output. So yeah.
Okay, thanks. Are there any other questions, Robin? No. Okay, so
I've got a quick question.
Yeah,
I'll just ask for super cool work, Robin. I was just curious about the scan durations and the signal to noise ratio, and sort of what you recommend is an optimal sort of approach for that. Like, do you expect that you'll get a better signal to noise ratio and better targeting if you were to use longer scans, for instance?
Yeah, I think that seems to be the case. I mean, I guess most of our research is kind of centered around the subgenual cingulate cortex, which has pretty poor signal to noise ratio, and also similar with the hippocampus, the work that we did there, or that Josh did, that where we just looked at the data. But yeah, I guess if there's a site that's that has a better signal to noise ratio, so something that's a bit more cortical, then maybe you wouldn't need the same scan duration. But at least for the SGC, we're thinking 15 to 20 minutes would be ideal for a resting state scan. But yeah, potentially you could go shorter. But also it can be useful to look at data sets like the Human Connectome project, and then just figure out retrospectively what is, how much variation is there from one scan to the next, what kind of scan duration do we need to be able to get a reliable target, for example, for a given site, or brain net.
Thanks.
Cool. Any other questions, Robin? No? Okay, so in that case, we might wrap it up. Thank you to all our speakers, and congratulations again on winning the awards. And we would encourage those of you listening to apply in the future as well for any of the upcoming rounds. Is there anything else, Hannah and Aaron? We might just say that we will have an AGM coming up soon, which we will post. So please keep an eye out for that. Anything else to add?
Just to the award winners, we'll be in touch very soon about your cash prize. So just watch your emails for that. And as always, just everyone, please let us know if there's anything you want us to be organising or doing. If you have any ideas for future webinars, anything like that, just drop us an email.
Yeah, I was just going to add, I think you might have emailed this out, Hannah, that there will be a talk on Thursday, just something informal, by a visiting researcher, and it will include some stuff around TMS. So that will be available online.
Awesome. Thanks, Robin.